minus 5 points for everyone who was late. No, what we're gonna do is we're gonna finish up that lecture that we. Oh no, my bracelet was broke. God dang, I like this one too. All right, what we're gonna do is we're gonna finish up the lectures from that we didn't finish last time. We kind of rushed the last little part, so we're gonna go back to it, and then the second half of this lecture today, we're going to give like an introduction to hematopoiesis, and then we'll finish up that lecture in the second hour, and then the third lecture will be on skin disorders. So all this being said is that you're only going to have 5 PowerPoints. It's a lot of rashes today. So you're only going to have 5 PowerPoints to study on this test, but it's like 6 hours worth of lecture. And then what I do is I do 5 questions per hour of lecture, so it's going to be a 30-question test for next week. So make sure to study. I will post a practice test though, which should help out a little bit. And some of the questions are going to be modeled in that same kind of fashion. You can kind of see the multiple choice levels, and you kind of gauge where you're at based off of that test. So, yeah, so just make sure to study the 5 PowerPoints, the bolded stuff, and the Halo stuff, and then we will, and then of course email me if you have any questions. I am going out of town, but we can still set up Zoom office hours if y'all have any like big questions or need something explained. Fair enough? Mm-hmm. All right, so let's go ahead and get started. So we talked last time about the background on immunity, and we went over inflammation pretty extensively and inflammatory mediators. And so what we're going to do is briefly just go over adaptive immunity and kind of T cell and B cell development. So hopefully y'all still remember that handout last time and what the cells are, and hopefully y'all also saw the handout for hematopoietic function that we'll be going over next time. If not, you can just give it a quick read during our little break. But so with adaptive immunity, what this is that it's activated after the innate response, and it's the whole purpose of it is to develop a greater exposure to the antigens so that T cells and B cells can be developed. And what these do is these target more specifically and more tailor-made responses for whatever the pathogen is. But importantly, like we kind of emphasized last class, is that you have these developed responses that target these things more specifically, but importantly, it doesn't eliminate anything. Antibodies don't kill anything in and of themselves. What it does is it recruits things that do kill them that are related to the innate arm. So what these do is you develop specific targeting of antigens, and it helps deliver innate responses in cells so that the innate responses better target pathogens, right? And so what you can see is that what you have an innate response to some viral load. If the innate response cannot deal with whatever the pathogen is, and the pathogen persists throughout it, the T cells and B cells will start to develop, and then those will help this innate arm reduce whatever that pathogen load is, right? So kind of went over what these different cells are, which are T cells and B cells. There are 2 different flavors of T cells, and that they're CD4 positive. That stands for cluster of differentiation. It's just a protein that is on the cell. It does something very important to the function of helper T cells, and then CD8 does something very important to the function of T cells or cytotoxic T cells, but y'all don't need to know that. They help with like co-receptor stuff. But importantly, what y'all do need to know is that the helper T cells, what they do is they recognize foreign antigen, and then they help activate the 2 arms, T's and cytotoxic T cells and B cells of the adaptive arm. While these cytotoxic cells, what they do is they actually do the actual effector function of killing infected cells and dealing with the cell-mediated responses. With B cells, there's a humoral response, right? These are the things that make antibodies, and they can develop further into plasma cells. There's this whole story of differentiation and concept called affinity maturation of the cells. It's really complicated with when you get to the nitty gritty of all this. But at the end of the day, B cells just develop into plasma cells, and then these professionally secrete antibodies to target whatever the infection is. For the humoral, if you have a humoral response, what we're mostly referring to is anything that's proteins that are secreted, which is mostly antibody responses, but it's also the innate arm is also that of the complement system, and those inflammatory mediators. That's also technically humoral responses. So if you're exposed to a pathogen, this causes an acute response, and then you activate T and B cells. If you are exposed to the pathogen again, the B cells can then go are ready to go, so that you have an immediate response to this. So what I'm saying here is that you have an exposure to an antigen, you have this response that we saw in the last slide, and then you deal with that pathogen. There's also the development of memory, to where you have certain cells persist that always remember that, and this is how long-term vaccines work, that or just subsequent infection. So if you have a second exposure to this antigen, whether it was an actual exposure, like let's pretend like you got a vaccine, you trained your immune system, you've developed that memory to where if you ever get exposed to that disease, you have an immediate explosion of your adaptive response, and that way your adaptive response is already already primed and ready and already working with your innate response to where you overcome whatever that pathogen load is. So all this to say is that your immune system deals with second exposures to antigens, whether it be from a vaccine or an initial time of getting sick, a lot better than the first time where it has to actually develop a response, develop those B. Does that make sense? All right, good. So yes, this is how vaccines work, right? So again, we beat to death by COVID vaccine examples, and so we'll just use that here, might as well. So you have a vaccine, this is what I'm saying. The first shot, your immunity actually develops. Sometimes you might even need a second shot. That's like a prime boost model are things. This is what COVID turned into. We didn't know it at the time while we were developing the vaccines, but right, where you get second shots, booster shots, long term. And what you're really just trying to do is that over time you can lose some of that memory, that those cells can wane away. Sometimes they're super long persistent. Like tetanus, I think, is like a 10-year persistence of those memory cells. To where you only have to get a tetanus shot every 10 years, something like that. I don't know. We have to look up the actual schedule of those vaccines, but sometimes they're super short, like the flu or COVID's turning into that, if it's not already, where you have first and second shots, and then to sustain that memory recognition of the cells to where you have constant protection over a long period of time.
This is probably a stupid question, but do you feel like the COVID shots were pointless, I guess, with the COVID vaccine cards and stuff like that? You feel like it was?
So, absolutely not. Where it's like because you have to look at like death responses. When you look at like pathogenicity of COVID over time, the way we overcame people dying, especially like old people and people who are more unhealthy, like don't have as good as circulation and oxygenation of their blood, they were vastly saved by the development of vaccines. That was like a public health miracle, the way that we developed those vaccines, and it drastically reduced the death rates and pathology of the vaccine.
COVID after I got the vaccine.
Yeah, so it's like right. So it's kind of a testament to this right here. It's like, yeah, you can still catch it, right? Because you're not necessarily have developed a strong immune system. Because also, what we're not talking about in this graph is that just because you've been exposed to antigen, do you really get a great development of your T cells and B cells? It's hard to say, right? Like you need a certain threshold of exposure to develop your immune system. And so if you don't have that, like you might have in the initial infection, or you might have with the first vaccination, right? If you don't have that, you need more. The immune system needs more exposure to the antigen to develop that better response. And so you just need to hit this protective immune threshold here, right? if the first vaccine was here and then the curve just dipped under here, you necessarily need that second, and that's very much the case for a lot of vaccines where you just didn't hit that threshold the first time, but then when you hit the second time, you get like this type of response, and you need like a third and a fourth shot instead of just one and twos, right? And so, it's exquisitely complicated and tailoring of the immune system, and you just need to really understand this stuff. I mean, this is what I have my PhD in, right? The immune responses. So it's like I understand all the intricacies and ins and outs, and I fully understand that when people don't understand these stuff, it's like it's very hard. It took like 5 or 6 classes for me to understand even the basics, right? So, and those are whole semesters and years of studying. So, but with that being said, to answer your question, for sure, the development of vaccine was crazy, and it immensely helped when you look at actually the study of the pathology of COVID. Yeah. Okay, so yeah, as far as the timeline goes, typically it takes about a week or 2 to develop these adaptive responses, and that's why sometimes whenever you get exposed to a cold, usually you don't start feeling sick for a few days in between. So like upon exposure and actual dealing with the symptoms, it's usually about a week or 2, right? And so that's because of these adaptive developments. All right, so B cells, they secrete antibodies. This is what a typical antibody looks like. You have the antigen binding fragment, which is just the part that actually binds to whatever the target antigen is. And you have this other fragment. This little fragment right here, you don't need to know this, but it's called the fragment of crystallization. This is what actually interacts with the immune system. And you can kind of see that in this picture right here, where you see that these immune cells can actually bind this, right, which you see in that inflammation. There's certain cells that can bind the Fc receptor of antibodies, and then that's how these antibodies help activate these things. Now, these are things like the innate response, right? So these are things like basophils that whenever they bind these antibodies, it helps deal with whatever the pathogen is. And this is why we say that the innate arm is really what helps deal with these stuff. Everything in this picture is dealing with the innate arm, maybe except for neutralization or precipitation, but that's like a separate issue here. And it can even be problematic in agglutination, where you're agglutinating your red blood cells and you're causing all these problems. But regardless, a major thing though is also complement activation, where you have activation of complement. That's also part of the innate response, where your antibodies are helping activate the proteins that deal with these pathogens, and it can cause things like cell lysis from your complement. So there are 5 types of antibodies, and I want y'all to learn. Y'all just need to learn in this graph. Y'all just need to learn the classes. Just be able to recognize it, right? Recognize that there is such thing as an IgA, IgD, IgE, IgE, and IgM. There's a whole bunch of stuff about like what they do, and there's kind of paragraphs under, or just like bullet points underneath them, where it's like what each specific class of antibody does. But I don't need to know that. But I just want y'all to know is that like, if there's a specific type of response, I want y'all to, like, and y'all see later on in the course, oh, this is dealt with an IgA response because it's a, whatever. Y'all know like what an IgA is. That it's like a type of antibody. That's all I really want y'all to know. But there's a whole bunch of characteristics about like how many are part of it and stuff like that. Y'all need to know that. Is that fair? So just make sure to, like, one of the questions might be, which of the following is not a antibody class? And I just pick a random letter of the alphabet that's like not on here, right? Like IGF, right? Yeah, right. So just be able to do stuff like that. All right. So as far as cytotoxic T cells, these deal with infected or tumor cells. These are cell problems with the actual cell inside themselves. Because the problem with the cell is that there's that lipid bilayer, the cell membrane that protects a pathogen, or that is your tumor cell. And so you can't just recognize it with an antibody, because the antibody doesn't have access to the things inside of the cell itself, right? And so what the immune system has figured out is that we just need to kill the whole entire cell. And so we have the rise of the cytotoxic T cell and also natural killer cells work the same way, that it recognizes that there's a problem with whatever the cell is, whether it's a tumor cell itself, right? We talked about like the cyathylation stuff and MHC, I think we mentioned briefly last class. Right? It can recognize that it's a tumor, or it can recognize that it's infected, and then it comes in and then it just releases a whole bunch of molecules that tells that cell to kill itself, and it goes through apoptosis. And so importantly, you might say that, okay, this is a cell-mediated response. The cell is killing it. This is like going against the paradigm that we've established of adaptive arms just tell innate arms to perform better. Well, importantly, what's going on here is that the cytotoxic T cell itself isn't killing it. It's not like a macrophage that's coming in and phagocytizing the whole thing and then digesting it. What it's doing is it's releasing granzyme B and perforin, and those are innate molecules that induce cell destruction, cell-induced cell death, right? So we're still corresponding to that same paradigm. Does that make sense? That perforin and granzyme B, those are innate molecules, right? And so yeah, so these cytotoxic T cells, they just come in and kill infected cells. So yeah, this is what I'm trying to get at. All pathogens get eliminated by the innate immunity, but the adaptive arm is crystal 2, aiding innate immunity to remove pathogens, right? So we have infectious agents / human relationships terminology. So this is some terminology I want y'all to be familiar with. So you have a host is any organism capable of supporting the growth of another organism, right? So if you got COVID, you were the host of COVID, right? And then there's even a host cell, right? That's because some of your cells didn't get infected by COVID, right? Whenever you get infected, your toe cells didn't get. All right, it's a bad example because of COVID toe, but your whatever your eyeballs. Yeah, I think your eyeballs were safe, right? Those cells didn't get infected. Those were not host cells. But your lung cells that got infected, those were absolutely host cells, right? Colonization is the act of establishing presence and a step required for infection. So that's just that's a pathogen coming in and then getting inside of you, but not necessarily infecting it. Right? You were colonized, but you're not necessarily infected. This is a big thing with bacteria because they can exist outside and not necessarily go inside of your body. So you're colonized but not infected. Infection is actually going in and then replicating within the host of another organism, and then their subsequent injury, right? So your commensal bacteria that are living in your gut, for example, those aren't actually infecting you because they're not causing pathology. Does that make sense? And then virulence is disease-producing potential of a microorganism. Is a pathogen virulent or not? There's a whole bunch of things that can get inside of you, cause some problems, but it's not really a problem. Like I can't think of any examples right now, but like if a virus gets inside of you and then causes you to cough a couple times, it's technically bad, but it's not very virulent, right? But if a organism comes in and it kills you, that's extremely virulent, right? So there's a whole scale of in between as well. So with microorganisms, you've got bacteria, fungi, archaea, and protists. Those are your major classes of microorganisms, and they can be beneficial or opportunistic, and they exist in a mutual relationship as well. So there's a whole bunch of microbiota on your skin, in your gut, and all over your body in certain areas, like on your eye, in your mouth, all over the place, where some of them are bad, but some of them absolutely don't do anything, and some of them are good, right? So we don't need to necessarily be germ-free insofar as if you make a disgusting face about me saying these sentences, you don't need to, right? Because some of the bacteria are perfectly fine and they do nothing, right? And some of them even help you, right? So you should be smiling instead. But so we have different relationships about what pathogens do, and that's an extensive area of study. So something like Staphylococcus aureus that exists naturally on pretty much everyone's skin, but it doesn't become a problem if it's outside on your skin, right? Because there's that dead layer of skin, and then there's protective oil things. This is why we say the skin is an organ of your immune system. That it's not until you get a cut or you have these microlesions where the bacteria get in and then actually cause staph infections, right? Where it's perfectly fine, but it's like when it gets inside your body, that's when it becomes a problem. And then we also have relationships inside of our gut where it's bacteria actually create certain really important vitamins like vitamin K um, that actually provide a benefit to the host organism itself. Um, and then they leave their normal sites. Now, the thing is that it's important that they stay in the gut. If they go inside your body and then start replicating and then start causing disease, you can get certain problems. This is like certain digestive problems that we'll talk about later in the course. Um, and then they can actually cause, but it's about like where they are, about what the relationship is. So if they're outside of your body, which is inside of the gut, right? It's and inside of the alimentary canal, it's fine and it's helping you. But if you get like some lesion to the gastrointestinal cavities and then it goes inside, then there are problems that can potentially arise, but not necessarily, because your immune system can also just fight back, right? With microorganisms, there's so you can have infectious disease where the host sustains severe pathological damage in response to the infecting organism. And they're opportunistic pathogens. The best example probably is, in my head at least, is what the virus I study called human cytomegalovirus, right? HCMV. There you go. So human cytomegalovirus, it's a virus that like infects pretty much 85 to 100% of the population after the age of 30, I think. So most people have this infection, right? And it absolutely can cause harm. The thing is that your immune system fights back very, very well against HCMV, but HCMV has figured out a way to hide from the immune system in this concept called latency. All this to say is that HCMV is probably even in everyone in this room's body, but your immune system quells it very, very well. The problem is that if you become immunocompromised, like if you get a transplant, like a heart transplant or something, you have to turn off your immune system to accept that graft, right? Or if you become immunocompromised from something like HIV comes in and specifically targets CD4 positive T cells. Well, that's a very, very important cell type that activates all your adaptive immunity. So if you target that cell type specifically, you get rid of all of your adaptive responses. You have a very, very weak immune system that relies solely on innate responses, and you're pretty much turning down the immunity. I mean, you saw those graphs, right? All those big curves about your immune responses, all those just don't exist. And so whenever you have an HIV infection or a graft problem, these opportunistic pathogens, whether you're Staphylococcus aureus or HCMV, they flare up and then can cause significant pathology and death, right? So in and of themselves, they're not very dangerous, but if you don't have your immune system, they become very, very dangerous. Does that make sense?
So can you your body fight that based on your overall health?
Like if you're running and eating vegetables and stuff?
Yeah.
Probably not, no. Because you need your immune system to really fight these things. Whether or not you get circulation through your muscles with oxygenated blood, it's not really going to do anything for something that's fundamentally infecting host cells and destroying them, right? Good question, though. All right, so we got different stages of infection. We kind of went over this. So we have colonization. That's the initial stage. And then there's like this incubation period where the pathogen is actually growing for a significant amount of time. Now, your immune response, even during this stage, this is when your immune system's starting to recognize and develop its adaptive responses, right? So the initial exposure to the immune system, that's when the immune system develops. You don't need a significant amount of incubation period. You just need a critical threshold of antigens to activate immune responses. So that's what the whole big deal about like what the dosage of the COVID vaccine was. There was a whole big drama about that, and they're troubleshooting different doses and doing dilutions within the population to see what's the smallest amount of dose we can give that gives a robust response. They're trying to figure out what that threshold is. Once the microbe crosses that, whether it be after the incubation period or not, that's when the adaptive response is going to be appear. Now, importantly, there's this critical threshold or clinical threshold um, that's where you actually start to see disease, right? This is where the bacteria has overcome any innate responses, and it's actually causing problems like coughs or fevers or whatever it may be, right? So then we have all these hypersensitivity disorders. So these are excessive or inappropriate activation of the immune system. So this is stuff like allergies, right? Like if you have a pollen allergy or an allergic response to peanuts or whatever it might be, these are hypersensitivity disorders. This is your immune system causing problems, even though the antigen that it's responding to is not bad, right? And so this causes damage to the body by the immune responses rather than the antigen itself. And so there's different types. There's type 1, 2, 3, and 4. And so I just want y'all to recognize different characteristics of each type. Questions like this will be, we have X, Y, and Z scenario. What is the type of hypersensitivity disorder? And then your options will just be 1, 2, 3, and 4, right? So with type 1 responses, that is like immediate hypersensitivity, and that's mediated by IgE. This is like an example about what I want y'all to know. Different antibodies, right? Y'all know that IgE is a type of antibody, and so you know that it's an antibody-mediated disorder. Type 2 is also a different antibody-mediated disorder, just not IgE related. Type 3 is the immune complex-mediated disorders, and type 4 is T-cell-mediated hypersensitivity, and we'll go over brief examples of this stuff. So type 1 is mediated by IgE, and it's stuff like asthma, allergies, and food allergies. You might have heard, I don't know if I go over it. Yeah, so you might have heard of this like cleanliness hypothesis where there's different areas that don't have near the hygiene that we have in America that get exposed to all kinds of pathogens that are. Wait, what? It's a very.
I was like growing up in that. Like 10 pounds. So I mean that wasn't a bad thing.
So yeah, not to take away from that. That's not really what I'm talking about. What I'm talking about is that no, you get. No. So it's places like third world countries that have a whole bunch that like they don't have clean water and they don't like well so the problem is that you've grown up and you've developed your immune system. So the thing is that these people that live in these countries or areas, they get constant exposure to these pathogens, right? And so their immune system's a lot better than ours, and so far as there's like a dance between the immune system and the pathogens that they're exposed to, that it's like the immune system wants to have certain things to react to, and it's like it's there because it's there for a reason. We've evolved this response for a reason. It's an evolution, right? Through and through, right? We've evolved these responses to do that, yeah. The problem is that in America, our cleanliness practices have gotten to a point where we don't see pathogens anymore. Like there's a whole class of parasites that we just completely eliminated from our airspace, pretty much. I mean, people get it and bring it back, but then it just goes away because there's no critical threshold, right? So the problem is that our immune system, we've eliminated the pathogens, a lot of them at least, not all of them. It's impossible to eliminate all of them. But the problem is the immune system still wants to react to stuff. There's not like a dance anymore between the immune system and the pathogen. So what our immune system does instead is it reacts to things that it's like searching for something to react to. And so it starts to react to things like peanuts and pollen and stuff like that, right? And so we get these, so places like America, we have a whole bunch of different types of allergies. But in places that have actual really poor hygienic practices, poor by our standards at least, which might be a better practice to be dirty, if that makes sense. They actually have better. They don't really have allergies because they're responding to the parasites and the microbes that are constantly presented to them. Does that make sense? Yeah, you can kind of look at it that way. Sure. That like not necessarily time, but it doesn't have the drive to. Yeah, but you're hitting that.
My family's from the Caribbean, and when I go there, I have to drink bottled water. I can't eat. Like, I have to drink.
Yeah.
'Cause it makes me sick.
Well, because
I grew up here.
Because your immune system's not used to it, right? And so it's like it's a foreign antigen at that point. That it's almost as if like by them drinking that water, they're constantly vaccinating themselves, right? Yeah. So yeah, it's a really interesting like phenomenon that's occurring, and so it's like and there's this whole hypersensitivities that happen because we just don't have exposure to antigens. Now, with that being said, there's also, I thought I had a slide about it, but I guess I didn't, or maybe it's later on the semester. There's also type 3 immune complex mediated disorders, and these are mediated by antibody antigen complexes, and these are things like serum sickness, where your blood is agglutinating and stuff like that. What is this? I don't know why I have this here. Oh, it just didn't show up. Antibody FC receptors. Okay, so with type 4 cell-mediated hypersensitivity, what's going on there is you have direct toxicity due to where is it? T cells, I guess, in this picture. So these are things like hepatitis B. So you can do different things about tuberculin test, and allergic contact dermatitis, rheumatoid arthritis is also part of this, where your T cells are actually attacking your joints, right? Again, it's a hypersensitivity because your joints aren't a pathogen that need to be attacked, but it's like something that develops over time. And we'll talk about that when we get to bone disorders more specifically. But with that being said, that's it for this slide. So yeah, there's going to be a couple more questions about immunity because we spent an hour and a half on it rather than just an hour. And we'll make sure you just study that. Any questions there? All right, so let's take a little break, and then we will. I'll get this posted, and we will pick back up on hematopoietic disorders.